So here, now, so someone is presenting. 1 minute, give control. Okay, it's fine. Polypeptide chains fall into several domains. This domain terms is now new to you. So the thing is, if this is one linear chain of polypeptide N and C, after folding, right? So the protein can be something like this. Complex structure. This is N terminus, this is C terminus. Or this protein, as I told you, we have more than 1 lakh proteins. So there are many defined types of fold, defined types of conformation present. Or it can be something like this. I'm just telling you something like this. Here N terminus, here C terminus. So if you see this, like this is a protein containing just one domain. That's one globular domain. Here, 2 domains. This is domain 1 and domain 2. Then what is the definition of domain? The domains means the fundamental unit of tertiary structure. And the definition is something like this. The polypeptide chain or part of polypeptide chain that can independently fold into a stable tertiary structure. That means if this protein is getting folded, like in first case, whatever I mentioned here, that just one domain protein here, like so here if you like cut this protein, like you have this the full protein here, like N to C, if you cut it from here, right, and then you want to hold this protein, it will not give you this structure. Why? Because the whole protein together they form this compact globule structure. This domain. So as a result of that, you need the full sequence. But in this case, in the next case, here, right, if you have this N terminus and C terminus of polypeptide, polypeptide chain, then you can see since you have 2 domains, you can cut here in the protein, okay? So that means if you cut this protein, still it will fall into 2 individual segments. So that means some proteins, they are containing some independent unit, full level unit, those are called domains. And domains are also unit of function. That means even in a single protein, say for example, a protein you have 2 domains. Maybe this domain they have different catalytic activity and this domain they have different activity. That is also possible because those are some kind of they can fold independently and they are maintaining their structure. So as a result of that, it is also possible some protein containing multiple domain with each of them they have different function also, right? So these are some kind of I would say some description of protein structure, nothing more than that. Now if you see here the quaternary structure, I already told you that one example is definitely whatever you know. This is from some textbook. You are not aware about this protein, so forget about this. Okay, so you can remember that RNA polymerase and as I told you before, like hemoglobin, all those things are example of quaternary structure, even antibody. But remember, like one thing you have to remember here, this quaternary structure, most of the time you have one polypeptide chain, another polypeptide chain, right? But those 2 polypeptide chains are not covalently linked. Here polypeptide chains are made up of covalent link because of peptide bond. But those 2 protein, when they are coming together each other, those are because of some electrostatic interaction, hydrophobic interaction. In rare cases, some covalent bonds are present, for example, in antibody, but we will discuss in a different class. When we will discuss about immune system, that time we will discuss about antibody, right? So now this quaternary structure, for example, in hemoglobin, we have 4 polypeptide chain. In RNA polymerase, how many we have? We have 5, but sigma factor actually lives, so even 4. 4 of them, they form the core RNA polymerase in bacteria, and that synthesize the RNA during transcription, right? So, but if we see even higher order, quaternary structure. This is one example. Hemoglobin, already I told you that out of those four, two are same. This is alpha, this is alpha 1, this is alpha 2. This just name beta 1, beta 2. So that means alpha 2, beta 2. They have total 4 polypeptide chain. Okay. And here in the next diagram, here you can see this is a very complex, it's kind of very, very higher order structure. The coat protein of a rhinovirus. Rhinovirus is some kind of cold virus that causes common cold. In case of virus, I think all of you know, we will take a different class about virus later. But in virus, like outside is a protein coat, like virus, they are not like kind of cell, but inside the protein coat, you have some genomic material, right? And now, if you see, the protein coat is made up of different types of proteins. But in this case, in rhinovirus, you can see like almost 60 copies of different proteins. They are staying together and they form the protein coat here. So this is some kind of superorder quaternary structure, right? So that's all about how you are actually going from from amino acid through peptide bond, you are getting the peptide and N-peptide, polypeptide, then you are getting alpha helix, beta strand and beta strand together, they are forming beta set, and then from tertiary structure, quaternary structure, and you might have many protein together, they are forming like very higher order quaternary structure. But now I have one here, like I would say few questions are there that I explained, or I tried to explain how alpha helix and beta strand or beta sheet form. But what is the rationale or what is the logic behind the formation of tertiary structure? How it will hold? What decide? Like what are the factors, physical or chemical factors that decides the holding of a protein? That we need to know, right? Here the question, the protein holding problem. Only thing I forgot to mention one small information. I believe all of you know. I should mention here. Otherwise, again I will forget. So, for example, dipeptide. When we say dipeptide, this is not related to this protein folding problem, but I forgot before. Dipeptide. Dipeptide means how many amino acids? Can you tell me? 2 or 3?
2.
So 3.
2.
Yeah, that's why the confusion there. Dipeptide means 2 amino acid, 1 peptide bond. Okay. Not that like 2 peptides bonds are there, right? Tripeptide means 3 amino acids, 2 peptide bond. So this is little bit confusing. That's what I'm saying. Okay. So this, just remember this. Dipeptide, tripeptide. So now the protein folding problem. So as I told you that from the linear chain or the primary structure, there are some chemical bases how they can form alpha helix and beta set. But then finally how they will hold to make a stable and functional molecule. You have to remember the ultimate thing. Whatever we discuss, the chemical properties or physical properties, that's a different thing. But the thing is, we are discussing about biological system. Finally, we want to convert the information present in DNA into a functional molecule so that it can function properly. Then in between, you can study like, chemistry, physics, statistics, whatever you want. But the final thing is you need a stable and functional molecule. If unstable or non-functional, we have very good machinery that will destroy the protein and those amino acids will be again recycled in our system. That is the good, good thing. But in some, in some cases, some protein folding associated diseases, there we have some problem, like protein misfolding happening very fast, and we don't have like or I would say our machinery cannot control or cannot degrade those protein too fast, and finally we actually like those misfolded protein get deposited inside the cell, and as a result of that many diseases actually appear for that reason. As example, I told you, like Parkinson disease. I believe almost all of you heard about its name. It's present in the last slide of this presentation. Huntington disease and some other folding related diseases are there also. Now, that phi and psi torsion angles are the only degree of freedom for the backbone, not the omega. Now, if you see here, so just think in a physical way that you have a polypeptide chain, some kind of model, right? So ball and stick model. Just you are trying to see those angle as well as those bond, and then find you have these. Those are the alpha carbon atom here, and not alpha carbon atom. Sorry, every atom, because otherwise you don't have this this angle here. So as a result of that, now if you actually try to rotate this thing in whatever you like, if you have some model, you can fold back something like this. But the thing is, you have to consider what are the side chains are also present nearby, because here you have to think now those knowledge whatever I started with, that those things now you have to bring here. Although you don't need to remember, some side chain they have negatively charged, like for example glutamic acid and aspartic acid. Okay, so they have negatively charged side chain. So as a result of that, if you think like here you have aspartic acid, here you have glutamic acid, they are very close by, they will repel each other, right? So maybe one glutamic acid and one lysine. Lysine is another amino acid that have positively charged side chain. That might help. So apart from this possibility of this phi and psi angle based on the bulkiness of the side chain, also the chemical properties, they are either they are hydrophobic or hydrophilic or polar, how they are interacting together. Those things also decide how protein will be folded, right? So now there will be some physical phenomena here. So this is, I just copy pasted from some textbook. You can see this. This is very classical thing, and it has been mentioned in every biochemistry textbook. That is called Levinthal paradox. I am not going into that. This is not the right thing. This is actually wrong, but that's why Levinthal paradox. So the thing is, what he actually calculated, that if you have 100 residue long protein, and you can see that each of those angles they can take 3 different conformation, right? So as a result of that, 3 to the power 100, that can be total number, right? That if the each position you can have like 3 different conformation. So now he calculated that if it takes 10 to the power minus 13 seconds to convert one structure to another, then how long how long it will take to fold the protein of 100 residue length. So this is, again, I am telling you, this is just kind of some theoretical thing, but that way protein never fold. So this is Levinthal paradox. But then we should know then how protein fold, because it is not possible to see whether this this fold is right or this fold is wrong. That is not not possible. The current understanding is that thermodynamic principle of protein folding. That means there is, and again, another scientist got Nobel Prize in 1967, most likely, Anfinsen, Christopher Anfinsen. So he actually demonstrated some classic experiment, and that discussed or that actually explained what is the major force or what is the rationale behind protein folding. Okay, and based on that, if I would like to say, I should say that protein folds within second, like millisecond, few second, not like minute or hour range. Okay, just after the synthesis through ribosome, right? You know how proteins are being translated. So then it can fold very quickly within second. So now here let us discuss like how protein folds. The 3D structure of a protein is encoded in its primary structure, the protein folding. So that how this primary structure, that means the one dimension of amino acid sequence of a protein, folds into its native three-dimensional conformation I told you before. And the thermodynamic hypothesis is the interaction between the atoms in a protein control the folding of the protein molecule into a well-defined three-dimensional structure. And finally, as I told you, that in 1950s, Anfinsen actually studies this protein folding, and he, as I told, he got Nobel Prize sometime in 1960s. I forgot the exact date, but that's not required to be remembered, right? So it's okay. So now this experiment is very classic one. I will suggest you, whoever present in the class, like pay proper attention. This is little bit complex, but that will tell you that protein can fold very fast, and actually protein folding depends on the sequential presence of those amino acid, the order of amino acid, that itself dictate how protein folds. And whatever this sampling of proteins we can see in today's world, that is because of like years after years of evolution, right? So that way you are finally getting some like stable native protein, which is also functional. So now here the experiment. So Anfinsen, in order to understand protein folding problem, you have to take a protein as an example. So what protein he took? He took ribonuclease A. This is one protein, RNase A. So from the name, you can understand what is the function of this protein. Just one of you unmute yourself and tell me what's the function of RNase.
Remove RNA.
Yeah,
Just to
remove.
cleave
Yeah,
the RNA from the
yeah.
transferring set.
Yeah, so as I told you, like we have polymerase, DNase, RNase. RNase means you are right. Remove RNA means it degrades RNA. It cleave RNA. It actually cleave the phosphodiester bond of RNA. Okay, so that's why we say RNase. All enzyme, biological enzyme, we start with AAC at the end. So now its function is to remove or not remove, its cleave phosphodiester bond in RNA, or it destroy RNA. So here this is just some ribbon diagram of RNase or RNase. Now some like few properties you should know before explaining this experiment. So here, this is just a segment of RNA. So this is, you can see into prime position you have OH. So this is an RNA, not DNA, right? And you have the phosphodiester bond. What happened if you add RNA and RNase together in a tube? Then RNase will degrade this phosphodiester bond, and as a result of that, it will be able to cleave the RNA molecule, right? So then why like Anfinsen took this enzyme? Actually, he took this enzyme or this protein very cleverly, because the assay is very like easy. Assay means like if he wants to study protein folding, like what you will see, like very briefly, I'm just trying to introduce. Uh, he took RNase, which is like properly folded native RNase, that enzyme, not RNA. RNA is the enzyme. Now he unfolded that protein. There are many chemicals you can completely unfold it, right? Even again, it will be almost like primary structure, right? He unfolded that protein. Now he was studying the refolding. So from the unfolded state to folded state, because see, inside the cell there are a lot of complexity. Many other things are happening inside the cell. Like at that time, it was not possible to study protein folding inside the cell. now people are doing, like because of advancement of many technique, people are studying protein folding inside the cell. But at that time, no. So what Anfinsen did, studying protein folding, you can say protein refolding. Why refolding? Because native protein, first he unfolded it. Then again he is trying to fold it. That's why refolding, protein refolding studies in a tube. So now what Anfinsen did, so he unfolded it and then he tried to refold it in presence of different chemicals to understand how this refolding is happening or whether it is at all happening. Then if the refolding is happening, then what will be assay? What do you think? Just from your own thinking, try to say that whether the refolding happened properly or not, how you will decide. Just one of you unmute yourself and try to answer based on the last 5 minutes, whatever I have discussed.
Whether it will again dissociate RNA or not.
Very good. That is the assay here. So that means you have RNAs in this, say for example, in this tube. Native, that means the normal RNAs. If you take little bit of enzyme protein from here, add in another tube that contain RNA, that RNA will be degraded. But if you denature this or unfold this RNAs, this enzyme, then it will not be able to degrade RNA. So again, refold this unfolded protein and then check whether it can again destroy those RNA. That means it has the catalytic activity, and the catalytic activity will be there if it folded properly. It is not random. It needs to fold very precisely. Otherwise, little bit, just one amino acid mistake, one problem, that will create the whole kind of lot of problem. So as a result of that, in this case, what he is assuming, that is right, that if you have you are getting the proper function again, that means your protein get folded. Right. So those are the background knowledge you need to understand this experiment. Now, this RNA, this protein is around 124 amino acid long. This is unnecessary information. You don't need to remember. That contain 4 disulfide bond. Disulfide bond, I didn't tell you before. This is the first thing you encounter, that what is disulfide bond. You might know from your previous knowledge in chemistry, but I will explain that. So now, here you can see Anfinsen treated his protein RNAs with urea that disrupts all non-covalent bond like H bond, ionic bond, etc. So as a result of that, no alpha helix, no other long distance interaction, only that peptide bond should be there. And beta ME, this is another chemical, beta mercaptoethanol, we commonly use in lab. So to break the disulfide bond, but now I need to first tell what is disulfide bond, right? Otherwise, it will create more problem. So if this is a protein, N terminus, and this is C terminus, I'm just ignoring this alpha helix beta sheet right now. Okay, so now you have multiple amino acids. So one amino acid out of those 20, that is called cysteine. This is, I would say, some kind of peculiar amino acid. This is the exception. So cysteine, they have, in their side chain, they have one SH group. Like one SH group. That's why I'm saying that they are peculiar. This is not normal. This is not present in any other 19 amino acid. So as a result of that, what happened? So before that, you have CH2 something there. I forgot also. You can also forget. Don't worry. So, but the end is SH. So as a result of that, that also helps in protein folding. How? So for example, this one, something like this. Here, see. Why? See, this SH here, right? And another SH from here. Okay, I'm just drawing in a little bit away. So this N and C here, SH and here SH, side chain. So maybe this one is the number 50 amino acid of this protein. This is the one amino acid, second amino acid going like this 50, and this is say 95, for example 94. But this 50 and 95 number of amino acid here position, they have SH group. If they have SH in oxidizing condition, what can be happening here, that this SH, like they can form a disulfide bond, covalent bond here, disulfide bond. So as a result of that, what will happen? So in oxidizing condition, it will make disulfide bond. So apart from the peptide bond, another type of covalent bond you are getting in a protein structure, and that helps in protein folding. I'm not saying that every protein they have disulfide bond, not like that, but many protein they have cysteine residue, and that can form disulfide linkage. Here you can see cysteine, and finally you get disulfide bond. Remember again, this is just 2 amino acid we are showing, but just 1 minute before in the whiteboard, I was actually trying to explain you how it helps in protein folding. It can actually bring two defined portion of a protein very close to each other because of this disulfide bond formation. Okay, so this information or this idea also you need to know in order to understand this experiment. So I'm now going into the experiment. So what Anfinsen did?
Sir?
Yeah, please.
Sir, which molecule does this oxidation reduction part? Like, how is it done?
So which molecule does this
Does
oxidation
this?
reduction part, right? In
Yeah.
the cell.
Yes.
In the cell. Yeah. So see, in the tube, like when I do this experiment, so this is very common. Almost every week, some of my scholars they do protein refolding. There we add some redox hormone, right? But inside our body, inside our cell, there are like what is called glutathione, oxidized glutathione and reduced glutathione, right? And cysteamine, hydrochloride, but those things you don't need to remember, right? So we have like
Sir, may I, sir, sorry to interrupt, but your voice is actually like it is turning into static.
cracking, right?
Yes,
Yes,
sir.
and now it's fine, but your video is also stuck. I think there's some internet issue.
I think internet issue. Here actually lot of rain from morning actually. Now it is little better.
Same here.
Yeah, lot of
Same
rain.
here.
Yeah, okay. Okay, I'm going even more slowly. So now, Soham, in order to answer your question, that inside the cell they have specific enzyme. Okay, so that helps in this oxidation reduction part. But before that, you have to appreciate one thing, that as I told you, if the position is like as I told you, like 50 50 position and 90th position. So 40 amino acid away. So how, even in presence of enzyme, how this disulfide bond will form? That is not possible, right? So as a result of that, somehow these two cysteines should be close to each other. Then enzyme will act on it, right? So then the question is how those two correct cysteine is coming into place. Because why I am telling you, so is the sound is okay, or it is still
Yes,
bad?
sir. It is okay now.
Yes, sir,
It
it's
is okay
fine.
now.
Okay. Then the thing is, see, if one protein, like polypeptide chain, they have just, two cysteine residue, just here one and here another one. Then there is no option, just it will, make disulfide bond. But the problem is many proteins, they have multiple cysteine residue. Say, for example, you have 4 cysteine residue. Then you have multiple combination. How, when the protein is unfolded, no, disulfide bonds there. Now, if this is the number 1 cysteine, number 2, number 3, number 4, then what will be deciding factor? Whether this cysteine will form disulfide bond with this or with this? That is a big question, right? So that is also it's a problem to understand, right? So let's see how Anfinsen did the experiment and what he suggested. So here you can see this is RNase and this is the N terminus and this is the C terminus, and this RNase has 8 cysteine residue. It's a lot of problem. 1, 2, 3, 4, 5, 6, 7, 8. 8 cysteine residues, right? And then what he did, he unfolded this RNase in presence of urea and added this reducing agent. Beta-ME is a reducing agent here. So as a result of that, cysteine are now in reduced condition, no disulfide linkage. Now what he did, we are showing just one molecule, don't consider one molecule, consider like 1,000 molecules there. Now oxidation with oxygen. This experiment is conducted in vitro inside tube. So oxidation with O2 under renaturing condition, right? So here renaturing condition means I'm just telling you what is happening. If this is the tube that contain denatured protein, that means it is very highly concentrated urea solution and it has beta-ME also and it has RNase. You have another glass here. That glass contain solution without urea. That is called renaturing condition. That means the denaturing agent is not there anymore. The unfolding agent is not there anymore. So in this tube, in my left hand, so another glass there without urea and also without beta-ME. In addition to that, you are actually adding some oxygen in this tube. Why? Because you are trying to oxidize those cysteine residue, that disulfide bond. You are helping to form the disulfide bond. So again, I am telling you, this is the unfolded state of protein. Here in this tube, you don't have protein yet, but you have solution without urea, without beta-ME, and in addition to that, you are providing some oxygen, right? Some air you are just passing. And now take some sample from here, from this tube to that tube. So as a result of that, what will happen? You are basically diluting the urea solution, like, so you need lot of urea to like unfold the protein. So whenever you are diluting here in this tube, in this glass, so as a result of that, that destabilizing force, unfolding force is not there anymore. So as a result of that, protein will try to fold. And during that time, also you are adding oxygen. So oxygen will also, like, help to make that disulfide bond. So as a result of this is called protein refolding. So this is called protein refolding. So now in this condition, if you see, when he did this experiment, he found that he is getting protein almost 100% active. That means through this process, he is getting active protein. That means again, that protein is active and it can break or it can destroy RNA molecule. So this is one part of the experiment. Okay, let me finish the next part, then you will ask me question. In the other half or the another experiment in the similar line, what he was doing, oxidation with O2 in the presence of urea followed by folding. That means I would like to say again here. So in this tube, I told you that you have denaturing agent or unfolding agent urea as well as beta-ME. But what you can do, you can take the sample from here, put in another tube, right? But in that tube, you don't have beta-ME. Reducing agent is not there, right? But you have that urea, but you are adding oxygen. That means in the unfolded condition, you are trying to oxidize those disulfide or those SH residue. Again, I am telling you, you are trying to fold the protein in presence of oxygen, but you didn't take out the urea or the denaturant. You didn't take it. Let's see what happened in this case. So in this case, what happened? Oxidation with O2 in the presence of urea followed by folding. That means you added first oxygen, then you are removing urea, right? You can use different tube or in the same tube, that's up to you. But the bottom line is, when he did the experiment, he found that only about 1% of protein are actually in native state or they are active. Only about, if that part is written here, you cannot see here, that 1% of protein, approximately 1% of proteins are actually functionally active, right? So now they have 8 cysteine residue in this protein. So now I will come into that why this actually that thing happened actually, this kind of complete different observation. In the first case, when you remove urea as well as you add oxygen at the same time, you have good protein, active protein, but here you are not getting. So that thing I will explain. But before that, I will ask you, can you answer this question? This is like you have to answer mathematically, not in a biological way. That why approximately 1% of protein are functionally active here. In this case, in the next experiment, whatever in the right side I explained just now, that why only 1% of proteins are active, or I would say actually in this experiment you will have around 105 possible combination, and that's why if 105 possible combination of different types of folds there, and out of that I told you protein is existing in only one condition, that is native condition. Just one is right, out of those 105 conformation only one is right. That's why I'm telling that approximately 1% are active. But tell me why this 1% is coming, why this 105 conformation coming. Tanmay, you unmuted, right? You will try, or someone else? Ronit, this is not biological question. If I tell you, then you will say, oh, I didn't able to tell. Easy.
So the question, can you say once again?
So the question is that when you oxidize first, you didn't allow the protein to fold. You have the urea in the solution, so that protein in unfolded state, but still you are forcing, you are trying to like forcefully oxidizing, you are adding oxygen. So as a result of that, you are not getting the properly folded protein, but you are getting 105 defined conformation of protein. And since you are getting 105 defined conformation, out of that only one will be correct, theoretically. So as a result of that, I'm telling you 1%. This is because if you see, we have 8 cysteine residue, right? So, I believe, good mathematics, like I'm not very good at that, but see, very simply, if you think in that way, you have 8 cysteine residue, right? I'm just drawing it in one protein, like these are the 8 cysteine residue. So if you take just one by one, the first cysteine residue, it has option to interact with, with seven different combination, 7. So then just, delete this. After trial and error, it disulfide bond from here. Then another one you have 5 option, 1, 2, 3, 4, 5. 7 into 5 into 3 into 1. So 105 different conformation, right? So that's why this is very, very classical experiment, and it is actually, you will find also in every textbook also. So the thing is, finally, this is from another textbook, but the overall idea is same, that how protein folds, the same experiment, and here some animations there, and here the calculation of 105 also in a more mathematical way. Why this 105 like combinations are like conformations are coming, but the overall the bottom line of experiment is why he got the Nobel Prize. It's C. I didn't explain that yet. The protein is constituted of different types of amino acid in terms of their chemical nature. So one unfolded protein, normally we unfold protein in presence of 8 molar of urea, very high concentration of urea, so that every other, like apart from covalent bond and the disulfide bond, everything is distorted there, nothing is there. So now, whenever I told you that take little bit of that unfolded protein and dilute it in a in a solution where you don't have this urea or any other denaturing agent. So in that aqueous condition where you don't have this denaturing agent, So what will happen? The linear chain, I told you at the beginning that hydrophobic side chain, they will be like afraid. They will be afraid to stay with the water. They will quickly form some kind of globular compact structure. This is called hydrophobic collapse. Those hydrophobic side chain, they will collapse very quickly. That is called hydrophobic collapse. That is taking only like milliseconds time, right? So some of you from physics background, like you are studying engineering background, if you are interested, study more about it, write me, I will send you good article about it. Okay, you will understand and appreciate better also, right? That protein folding, like some of your, like in physics department, some professor also there, they work on this protein folding issues. So now the thing is, this is called hydrophobic collapse. So that means whenever you are taking away that denaturing agent, they cannot stay in the water, quickly they fold together. So here, if I go here in this slide, you will get it. So hydrophobic core formation is the first step of protein folding, and then those hydrogen bonding and long distance electrostatic interaction, all those things will happen. So now, how to create such hydrophobic core? You can see here, this is the kind of segment of a polypeptide, or you can consider this is a polypeptide, and it is in water solvent, right? Solvent is water. Those are the water molecule. So as a result of that, this yellow colored side chain, those are actually hydrophobic side chain, right? So as a result of that, you can see they can quickly form this compact structure where these hydrophobic residues, they are stacking together, so that multiple hydrophobic residue, they are closely attached with each other. So as a result of that, they are little bit buried inside the protein structure. And some other hydrophilic as well as charge residue, they are facing water molecule, they are staying outside. So that way, that is the major driving force of protein folding. Okay, I'm sure that you might have problem. Some of those problem I like, I can try to address, but because of time, I will not discuss in more details. You can write me, because this coating, refolding, unfolding, this is one of my routine work in the lab. Like my PhD scholar, like almost every week they do the same thing. So quickly, if you have question, like you can ask me, then I will go to the next slide. Okay, fine. So time is also getting close, so I'm sure that you are also in rush. So I'm going into the next slide. This is the last slide. Then we have 3-4 slides, but that is not for exam. That is for some disease example. This is what I want to tell, that can we predict protein structure from sequence? That is the question, because as I told you that how protein folds, that's a mystery, right? What I explained, that is the basic, very basic outline of protein folding principle. But then just, every protein, like say for example, one protein, just, one mutation that causes like that completely, convert that protein into a different, different form. And that is, you cannot explain all the time. For example, nowadays you can see in the newspaper in article that the mutation of spike protein in corona, like COVID-19 viruses. That's one mutation. How this, completely impact the protein structure. This is almost sometimes beyond our imagination. Sometimes we can understand, but sometimes not. So as a result of that, and also in order to understand, like everything happen, any mutation, any changes happen, then again you have to solve the protein structure, right? That is also time consuming, and also you need like good expertise to solve structure, either X-ray crystallography or NMR. But then what is the another way around? So people are actually trying throughout the world that whether you can predict nicely the structure of a protein from the sequence itself, because based on the Anfinsen experiment, the order of amino acid sequence, that dictates the protein folding. That actually dictates that how this hydrophobic collapse will happen. So as a result of that, you can see that Microsoft, Google, and all of those things, they have research team. They are trying to understand this prediction, or they are trying to improve this prediction, that sequence to structure prediction. And there are some kind of competition that is, you can see that every year that is happening, and this is DeepMind's AlphaFold 2. This is, I will not ask you any question from here, but this is just, if you get interested, you can search a little bit later and you can actually read about it. So this is, mostly like, deep learning program, which is, those kind of computational thing, which I am unaware, but if you are interested, you can actually go through it. And the bottom line is, from sequence to structure, how you can predict. So if you can predict very nicely, then all the time you don't need to solve protein structure experimentally, right? At least at some point of time you can predict nicely, and based on that you can proceed some of your work. But later on you can again, again you can experimentally prove that thing.
So,
That is okay. Yeah, yeah.
so
Tanmay.
In Alphensen's experiments, they had cysteine residues.
Hmm.
So the structure was based on that. So what about a polypeptide chain which does not have cysteine residue?
Yeah.
So will it work?
Yeah, Tanmay, good point. So without cysteine residue, a very good point actually, that without cysteine residue, mostly that protein will not face that kind of problem. What actually Anfinsen actually observed. You are absolutely right. So that's why most of the protein without cysteine residue, normally they are very good. That you unfold it and you refold it. Refold means you are just taking away those denaturing agent. Then you will see that protein will be in good conformation, right? But still, I'm not going into that details. But only that's taking away the denaturing agent sometime not enough. In our system, in our body, in our cell, we have different protein folding modulators. Those are called chaperone, right? I'm not going into that details, but some other protein, they help or assist in this folding. But normally in physical chemistry lab or physics lab, whoever study protein folding in vitro, normally they ignore this thing because they are studying like protein folding kinetics and all those stuff with some very standard protein, like they normally buy protein from some company. But most of the biology lab, so for example, even I would say in my lab, what we study that is more in context to biological system. Sometimes we study protein folding inside the cell also, right? So in that time you have to consider all those things, right? But mostly like, you are just studying the folding kinetics or the folding and those things, then it's fine actually. And that time people normally take protein without cysteine. So what you pointed out, that is right. Without cysteine, it will be much more straightforward. Only thing is that hydrophobic side chain should be packed properly so that the hydrophobic core can be formed, and then in the periphery you should have more polar and charged residue. So
Yes, sir.
now it's almost done actually, and time is also over. So this AlphaFold 2, this is just, if you are getting interested at some point of time, you can just search. And these things is mostly informative. I will not ask you any question related to this disease, but there are many diseases which are happening because of protein folding problem. This is called misfolding of protein. One example is cystic fibrosis. This is related to some transmembrane protein, right? So then the next one is Alzheimer's disease. That is because of some amyloid plaque or some aggregated protein which are deposited in the brain itself. So that causes, problem, and this is very common disease, Alzheimer's disease. And another one is Parkinson disease. Mostly, it leads to shaking, stiffness, and difficulty with walking. This is again some protein folding problem. Unfortunately, as I told you that in the beginning, that G.N. Ramachandran, he contributed a lot understanding phi psi angle, and that helps to understand protein folding. But he suffered like last few years before his death from this Parkinson disease, and in 2001 he passed away. So anyway, this is all about proteins. This protein folding diseases, I just went through it, so I will not set any question from the diseases, but you should go through it to get some idea, right? How protein misfolding can cause this disease and what is the reason why they are forming the disease. Even those 3 diseases you can search in Wikipedia, and little bit, at least first 2 paragraphs you can read, and I'm sure whatever background knowledge I have provided you today, that will be sufficient for you to understand. Okay, so thank you very much. So I'm done. Yeah.
So the three diseases are Alzheimer's, Parkinson,
And cystic fibrosis. There are many actually. Those things will be in the slide. So, like sometime when we used to take class in the classroom, we used to teach those because in online I'm like taking more time to explain. Right? I cannot use proper board and that's why. So, but anyway, but since I'm not discussing in details, that's why I'm telling that we will not set any question, but you should read. So you will get some interest actually. Another one.
Find some very good resources like something that everyone can read. You can post it in the Teams channel.
Yeah, sure. Another disease is called prion disease. For prion, that is another misfolding disease. For even just for prion disease,
mad cow.
Hmm?
Mad cow disease.
Yeah, mad cow. This is similar, like prion. So for that also, scientist, his name is Prusinar. So he also got Nobel Prize. So because of this mad cow disease, whatever you were saying, yeah. So I will post some of those good material in the MS team. But again, I'm telling you, this is optional. Like whoever you are getting interested, you should go through it. Okay. Thank you very much.
Sir, on which email ID we have to mail you?
Rajesh?
On which email ID we have to mail you for doubts?
Okay, so please, I forgot. So Rajesh, so I actually one email I have with me about your translation question, so that I will answer that. And those things are okay, like a little bit of more details. And for any query, you should email me to my iitkgp.ac.in, not the Gmail. Okay, and for only for submitting like assignment, you should use that Gmail ID. Okay.
Yes, sir.
Okay. Thank you. So you can leave and you can take rest. Yeah.
Thank you, sir.
Thank you, sir.
Thank you very much.